All right, in lab today we're going to be using pennies to represent the genes of an individual. So you know that a penny has a heads side and a tails side, and so we're going to be representing heterozygous individuals, that is, individuals that have two different kinds of alleles, a dominant allele and a recessive allele. So in our model, we are going to have the tails side represent our recessive allele. Remember, recessive alleles we usually show as a lowercase letter. I'm using a lowercase s, and I've made it cursive because sometimes when letters look really similar as capitals and lowercase letters, it can be hard to distinguish what you've written in your Punnett square if you don't turn one of them into cursive. So I'm going to highly recommend to you, anytime letters look similar, that you use the lowercase written in its cursive form so you can tell them apart. So again, the tails of our coin will represent the recessive allele, which in this case is the gene for long, big toes, and the heads side of our coin will represent the dominant allele, which in this case is the gene for short big toes. And each time we flip the coin, we are modeling Mendel's law of segregation, which says the parent can give one of those two alleles, but never both. So you're going to have two pennies, one penny to represent each parent, and you're going to flip them together to represent the formation of gametes, and then those gametes, we're representing them coming together in the formation or in the process of fertilization in order to show the creation of offspring. So let's look at what that might be like. Now, a really important key is that you need to actually flip your two coins. Please don't just toss them straight up. Make sure they're flipping each time that you flip them up in the air so that it's really random. You want to actually do this 100 times randomly, flipping both coins and looking at the outcome so that we can see if a Punnett square really is a good model for what actually happens when you have a random event like a coin flip. So imagine that I've flipped my two coins and I get this result, heads with heads. So we know that heads represents the dominant allele, so I have the genotype big S, and the phenotype for that individual, since it has two copies of the dominant gene, would be a person with short big toes. So then I pick up my coins and I flip them again, and the second time around, oh, it turns out I get heads again. So once again, my genotype would be big S, and my phenotype would be short big toes. So I'm going to do that 100 times. I've modeled that for you just 8 more times for a total of 10. And you can see the results that I got as far as heads and tails and what that represents for the genotype and what that represents for the phenotypes as well. Now please notice if I get a tails heads, that gives me the genes little s, big S, or if I get the heads and then a tails like this one, that gives me a big S, little s, but those genotypes are equivalent. So typically we always write the capital letter first and the small letter second, the lowercase letter second, just so that they look the same. So I've written it in the way that these coins were flipped here, but normally we would actually always write the dominant gene first and then the recessive allele second. So good luck. Once again, please make sure that you are actually flipping your coins each and every time. For a total of 100 flips, and you want to make sure that you keep track for each of those flips what the genotype is. You can always go back and figure out later on what the phenotype was.